minute past. So we're going to go ahead and kick this off. Again, this is the Genetic Ambassador call for Mountain States Regional Genetics Network. This is our quarterly call, and this is our July call, July 2021 call. And today, I am excited that we have a guest speaker joining us. We're going to start off with some introductions, and then we have a couple other topics that I just want to give some updates and get some feedback on. We have our Red Flags for Genetics handout we'll talk about, and our Genetics Explainer video introduction that I want to share with you. And then hopefully there'll be a little bit of time at the end for some discussion on future projects. But with that, we always start these calls with one of my favorite parts is introductions of everybody on the call, because these are really meant to be ways for our genetic ambassadors across our 8-state region to connect with one another as well as with Mountain States. So with that, as you know, in good genetic ambassador fashion, we always have a question to answer as well as we do our introductions. So when you introduce yourself, if you could just say your name, your state that you're representing, and then this question comes from some of the topics we're going to discuss later in the hour, which is what was the most difficult genetic word or concept to comprehend or understand for yourself and your loved one on your own family's genetic journey. So with that, I have at the top of my list here, Daphne. Are you back? Daphne, do you want to introduce yourself?
Okay, I'm Daphne Fulton. I have a grandson who has vascular Ehlers-Danlos syndrome, and I research health-related quality of life issues with people with rare genetic disorders. Okay, so I had actually worked in genetics for quite a while before I went back to school and got a doctorate and became a professor in public health. But the most difficult genetic concept for me was that a de novo mutation actually happened to us. What are the risk factors? What are not the risk factors? Nobody knows. And the fact that it happened to us, my daughter is a nutrition nut. I mean, just all the variables that way. It was kind of like, holy cow, how did, it was the de novo mutation happened to us with no known risk factors. So yeah.
Thanks for sharing, Daphne. I appreciate that. Next up, I have Hanan. Would you like to introduce yourself?
Hello, everyone. I don't know if you can see me. Sorry.
We can see you.
Oh, okay. Hi, I'm actually right now at the Children's Hospital with Miriam, my daughter, at an appointment. And I have some time, so I thought I had scheduled 2 on the same day because I hate the trips here. It's not that far, but I just, driving, traffic is bad usually. So I'm waiting for the next appointment. I thought I should jump on the call and join you guys. I'm Hanan Dawood. I'm a mom to a 3-year-old, chromosome 18 diagnosed child. And I'm actually in school or going back to school to be a genetic counselor, hopefully. And I have some background in trying to go to nursing. But other than that, I just learning as I'm going with my daughter's diagnosis. I am a mom of 5. She's the 4th child. And so due to a little bit of the nursing educational background, I can't think of one concept or one word that I thought I struggled with, but I kind of just learned everything as I went. Let's see, I really can't think of one. Maybe it's because it surprised me, but maybe I can think of one later. But yeah, so far, like I said, everything is based off of learning as I was going, and everything else just came, well, the diagnosis came out once, but I learned everything as I went. So that's pretty much what I wanted to share. It's so nice to meet you all.
Thank you, Hanan, and thanks for joining us in between appointments. We appreciate you hopping on today and hope the rest of your appointments go well. And feel free to email me if you think of even a word that maybe you didn't struggle with because you had the background for it, but maybe I know you let me know that you're doing some work nationally with some special needs groups. So even maybe other words that other families might struggle with in genetics. So if any of those come to mind, shoot me an email.
Definitely will. Thank you.
Thank you. Thank you. All right, next on my list, I have, let's see, Jamie, do you want to introduce yourself?
So let me turn my video on. There we go. So I'm Jamie Stefanski. Sorry, I'm cramming down my lunch before I have to pick up the kids. And it's not a word, but it's just a general concept that in not only genetics, but in all of medicine, as a parent and as a professional, as a PT, having worked in hospitals and clinics and such, that the rampant invalidation that medical professionals have with their patients and their lack of willingness to explore further something that a patient might bring up or a new condition. So I've run into that, like I said, as a parent, as a professional, and also as a genetic navigator now. So that's just a general concept that I'm surprised and struggle with that across the board.
Thanks for sharing, Jamie. And I don't know if you said it, but you're from Colorado, right?
Yes, I'm from Colorado, and I have 2 kids with Telo-2U-Verfong syndrome.
And Hanan, I don't know if she said either, but she's from Texas as well as Daphne. All right, so next up I have Lori, another genetic ambassador from Colorado. Lori, are you there? Open. I think we're getting a little bit of background noise. Can everyone that's not speaking mute their lines? Thank you. Is Lori there? Oops. If not, we'll go on to Lourdes. Would you like to introduce yourself?
Today I parked there again. But do you think if I show them my little...
Sure, sorry. My name is Lourdes Torres, and I am a parent educator for a community research center here in El Paso. I am the genetic navigator for Texas. What was most difficult for me to understand is when the doctor told me that my daughter have a genetic mutation called de novo, and we didn't know what was it, and we have difficulty understanding in general the genetic concepts. But I remember when they told me it's a de novo mutation, so we were sad. Then we begin trying to look for information about it. Now we don't feel that lost anymore. So I'm glad to be here. Sorry, I am on a capability for youth with disabilities right now. So in a moment I need to turn off my camera, but I'm here.
No worries. Thank you, Lourdes. We appreciate you. Thank you. Thanks for being here. All right, and next on my list, I have, well, let's wrap back. Lori, did you want to introduce yourself? I don't know if you can hear us now.
Yeah, hi everybody. I'm Lori Weiss from Denver. I'm actually at the hospital right now getting an infusion, so I'm going to listen in on mute. But thank you.
No worries. Thanks for being here, Lori.
Okay.
All right, and then next I have on my list Elvira. Elvira, would you like to introduce yourself?
Hi, my name is Elvira Dennison. I live in Nashville, New Mexico, and the Navajo Nation. I am a family mentor for Navajo Family Voices. I guess the question of most difficult word, well, it wasn't mainly difficult. It was just, it's not common for that word to be used around in genetics, not knowing what genetics was really about until I did my research with what genetic means with different types of broad meaning to it. I had an uncle who had Down syndrome, and I took care of him, and not realizing that there was a lot of resources for him that we didn't know of. And that's probably something that a lot of people or families not aware of, because we have limited resources out in our area and not much of internet that we have available here in our area. We don't have trainings or conference nearby to talk about the definition of genetics. That's not available out here. So being a part of the Mountain States Genetic Network was an eye-opener for me. Just kind of giving updates about this program to other families that are interested in it. And I've seen people who are probably following my part of my Facebook, and I did believe that I did connect to quite a bit of people.
Thank you so much, Elvira, for sharing that. I appreciate your honesty, and I appreciate that you're here with us. And it's always good to remember that we need to sometimes just start with the basics of the word genetics, right? It can mean a lot of different things for a lot of different people. So I appreciate your perspective on that. Thanks for sharing. And next I have on my list, Winton. Winton, do you want to introduce yourself?
Hi, yeah, hi everyone. Winton Wood. I live in back and forth between Albuquerque, New Mexico, and then a little village up north in Taos County. And I am mom to 2 children. One of my daughters, a teen, has Down syndrome and is medically complex, and she has autism. And then professionally, I work at University of New Mexico's Center for Development and Disability. I'm a case manager for individuals who are on the medically fragile MEVIA waiver or the developmental disabilities MEVIA waiver. I also am a coordinator for the supports waiver, which is a waiver that we have in New Mexico. It's a small waiver for those that are on the wait list to get on the full developmental disabilities waiver. Nice to be here.
Winton, was there any words that you wanted to highlight or concepts that were difficult in your journey?
I'm sorry, yeah. Well, if I go back to the birth of my daughter 15 years ago, I would say almost all words. Mm-hmm. It has to do with the IEPs, special education, IDEA, all the acronyms. You know, I would say maybe my pet peeve, although I try not to have pet peeves, is when we use acronyms as professionals and just kind of forget that not all of us know what the acronym is, what it stands for, or that, like me, I forget every time. I need to be reminded. So I guess I would say it's not really a word, but I would say acronyms.
That's a great one. Alphabet soup sometimes, right? Thank you so much for sharing that. All right, next I have Hannah. Do you want to introduce yourself?
Yes, thank you, Christy. I'm Hanan Barnhart. I'm in Nevada, and I am currently the genetics navigator for Nevada. And I agree with Winton, actually, that acronyms sometimes gets in the way. I've worked in the healthcare field myself, and when I was new to genetics, everybody's saying, oh, you know, you can do a CMA or a WES, you know, you just never know. And I'm like, well, what the heck is a CMA or a WES? So chromosomal microarray, whole exome sequencing. Yes, just all those acronyms. And English is actually my second language as well. And so I firmly believe that when we are speaking to the general public and even with each other, you know, we need to realize all those things that sometimes another person doesn't know about. And so we need to be cognizant of that when we're speaking with each other.
Thank you so much, Hannah. I appreciate that perspective. Always a good reminder. And I'm gonna read, I just noticed in the chat box here that Brandy introduced herself. She had told us at the beginning of the call she doesn't have audio. So Brandy's from San Antonio, Texas. She works at the Children's Hospital of San Antonio Laboratory Department, where she does a lot of genetic testing there. And she's interested in buccal swabs and newborn screening collections, less invasive collections for patients. All right, and next I have on my list, Mary Jo. Would you like to introduce yourself? If you're talking, Mary Jo, you're on mute. We can't hear you. Okay, I'm gonna skip on then to Casey. Are you still on? I saw your name pop up, and are you still there, Casey? Maybe not. Okay. And then I have two phone numbers. I have one from 512 and one from 724. Would either one of those people like to introduce themselves?
It's 724 is me, Jamie Christie, because I had to switch over to phone.
Okay, no worries. Thanks, Jamie. And anyone from 512 want to introduce themselves?
Hi, this is Melissa Fox.
Oh, Melissa. Good to hear from you. Go ahead.
Yes, I'm driving. I was at a Celebration for Life, one of my friends and coworkers who passed in January, and you know, they waited so that we could get past the not supposed to be gathering in large groups, and here we are back in Texas wearing masks again. Not by the governor's decree, but you know. I am part of the Texas Mountain States team, and I have an adult son with Down syndrome. I work for Texas Parent to Parent. I'm sorry, I'm driving and not able to focus, but I think I hit the highlights. Thanks.
Perfect. Glad you're here, Melissa. Thanks for calling in. I'm sorry to hear about your loss, but glad you're able to congregate and celebrate your friend's life. All right, did I miss anyone that did not get a chance? Oh, Heather Schichtel. I see Heather just joined. Heather, do you want to introduce yourself?
Yeah, hi, I'm sorry I'm late. Heather Schichtel, I'm part of the Colorado State team and run a nonprofit here called Miracles for Mito. We focus on families impacted by mitochondrial disease.
Perfect. And did you have any, or we're answering the question, what would any difficult genetic words or concepts to comprehend when you were on your genetics journey? Are there any, and you don't have to tell us now, if you want to think about it and pop it in the chat, you can do that too.
Odyssey.
Odyssey. Oh, good one. All right. All right. With that, let's see. We do have Cody on here, so I want to make sure he introduces himself, and then I will introduce myself and then introduce our guest speaker. So go ahead, Cody. Would you like to introduce yourself?
Hey everyone. I think I've met a number of you, but I'm Cody. I am the state team's coordinator for MSRGN, or if we're not using acronyms, the Mountain States Regional Genetics Network. I always had trouble sort of with chromosomes and how like what is and what isn't a chromosome in cell and DNA replication.
Good one. Thank you. Thank you, Cody. All right, with that, I'm going to go to our next slide here. So I am Christy Weiss. I think I know almost all of you, but if anyone's new to the call, I just wanted to put a name with a face. So I am the projects manager for Mountain States Regional Genetics Network, and lead these genetic ambassador calls. And one of my favorite highlights of our quarter is getting us all together. So glad you're all here today. And I'm really excited to introduce our guest speaker, and I will let her give more introduction of herself, but Dr. Nara Sobrera, I don't think I didn't get it all right, but I'll let her say it correctly, is an assistant professor of genetic medicine at Johns Hopkins University School of Medicine, and she leads a project called GeneMatcher. And I'm trying to remember how I came across this. I think it was through some of the ACMG listservs that I'm on for the American College of Medical Genetics, and spoke with her. I think it was back in the fall, and they were awaiting funding for their new round, and so they've received that. And so I asked her to come on today to tell us a little bit about GeneMatcher and the program that she runs, and it's in conjunction also with a group in our region, which is Baylor College of Medicine. So I will let Nara take the stage, and I will stop sharing my screen if she would like to put any slides up. Nara, are you still with us? There you are. You're on mute.
I'm muted. Sorry.
There you go. That's okay.
Doesn't matter for how long we have been using Zoom, still get to talk when I'm muted.
No worries, no worries.
No, thank you, Chrissy, for having me here today, and thank you everybody for being here. Like Chrissy said, I'm at Hopkins and I'm a clinical geneticist, and I do research also on rare Mendelian diseases. And that project that I'm going to talk to you today actually started from us investigating the genetic causes of patients with rare Mendelian diseases. Project that we started. That we started 10 years ago. That's called Baylor Hopkins Center for Mendelian Genomics. And it was an NIH-funded project that had the goal to sequence, each center had to sequence about 1,000 individuals with rare Mendelian disease per year, trying to find the molecular cases, the molecular basis of these cases. So let me just share my screen with you so I can put some slides up. And actually, as a clinical geneticist, my focus on the lab, beyond all the rare Mendelian diseases, is in a few diseases that are characterized by mosaicism, and explaining mosaicism and how mosaicism is not inherited or can be inherited. It's very hard sometimes to me to get the patients to understand that. So we were all talking about what's hard. Some things are hard for us to explain also, not only for you to understand. So I just thought I would say that. But I want to talk to you about GeneMatcher. GeneMatcher is a project that we started 2013, actually came alive, and it came alive because we were as part of this project doing a lot of whole exome sequencing. And I think many of you have heard or have had someone on your family or even yourself to do a whole exome sequencing, trying to find the diagnosis for the Mendelian disease that are affecting your families. So when we do the whole exome sequencing and you receive a report, many times you get a report that is saying negative. But what's not telling you is that there are many variants that were found and that were analyzed. It's negative because we could not actually make sense of any of them and connect to the disease that's being investigated. It's not negative because there's nothing there. So GeneMatcher comes exactly to try to solve these negative cases. It's a way to us to try to find out in there what could be relevant for the patient. So as I said, 10 years ago, we started using a lot of whole exome sequencing on research and later in clinical setting to do this investigation of to find the diagnosis of our patients. And it generates a lot of data. So just to have an idea, every time we do a whole exome sequencing, that is just 2% of our genome, we come up with about 80,000 variants, 80,000 variants. So it's a lot of variants, and we have to go down from 80,000 to 1. So it's a huge amount of data, and we are trying to make sense of this data, trying to find for novel disease genes and also novel variants that are causing the disease that we are investigating. Just to have an idea of how it has been in the last 10 years, how this whole exome sequencing and now more and more whole genome sequencing is helping us in this field. This here is a graph that comes from data in a database called OMIM. And this is a database that tries to have a page for every rare Mendelian disease that's out there. And every time a disease is connected to a gene, this relationship is also found in that database. So here on the purple bars are the number of phenotypes that have been entered every year in that database. So these are diseases that nobody knew existed. But now, because we found few patients with the same phenotype and we found the gene that caused this phenotype, can be recognized as a diagnosis. And then you see in the orange bar the number of genes that actually have been entered in this database as a gene that is known to cause a disease. So more or less every year, 200 new phenotype gene relationships are entered in this database after we started using whole exome sequencing 10 years ago, more or less. And these numbers went from 2005 at 11 years ago, we had the number of genes that were associated with a disease in this database was 2,500. And now, 11 years later, thanks mostly to the use of whole exome sequencing and a little bit of whole genome sequencing, it's 4,500. So these are 4,500 genes in nowadays that we can actually look at and know that if there's a pathogenic mutation in one of them, it will cause a disease, and try to correlate with the disease in our patient that we are caring for. The 12.5 is a percentage among the total number of genes that we have in our genome. So we predict that we have about 20,000 genes in our genome. So now, in nowadays, we know of 4,500 of them being associated with a disease. It's 22%. So we still have a lot to find. What are these other 16,000 genes causing when they are mutated? But okay, it's helping a lot. How can it help more? So I told you we have all this data when we find when we do the whole exome sequencing of a patient, and we want to find what's the novel disease gene or the novel variant in there, causative variant. One main step that became clear we would have to work on to solve these cases was data sharing. I cannot just have my group of patients in my lab that I'm investigating, do the sequencing of them and trying to solve them just in there myself. I have to actually get out and look for patients that are similar, to look for patients that have mutations in the same genes. I have to be talking to people not only in US, but in every other country that I can, because as you know, many of these diseases are very rare and there may be only 10 patients around the world with the same disease. So if I'm just focusing on one city or one country, I'm not going to be able to solve the case for my own patients. So we clearly started talking about data sharing of whole exome sequencing data 10 years ago. And these questions that are here, some of the questions that we discussed 10 years ago and that we are still discussing. So neither one of them have been solved perfectly. Everything we do, we try to address these questions in the best way that we can. And we are always changing and trying to improve. But as of now, they are not solved in a perfect way. What kind of data to share, right? Are we talking about sharing the whole, like when patient receive a report, it's just a report. If the patient wants the data, what are we talking we are going to give to this patient? What does giving the data to the patient means? The data has many steps that it goes through. And depending in what step you look at this data, it may mean nothing, even for me, can you imagine to a patient? So, and even if we are sharing among ourselves, what makes sense? Are we talking about the raw genetic data? Are we talking about a more processed genetic data? Are we talking about the phenotypes, the features? So all these things came to discussion, like what we are talking about that we want to share, how to share, where to share, with whom to share, how to ensure that the researchers, and this was a big problem because researchers go through a lot of trouble to get samples that they can use on their research. And they felt more 10 years ago than now, thankfully, that they could not just give up on the data to the entire community, because then they would kind of like lose all the work that they had done if someone just got that data and found the answer and didn't include them on the publication, let's say. So that was a big step on data sharing. Most of the patients were like, yeah, you can share, but the researchers who had actually collected the samples and done the sequencing, they were like, no, I can't share, because if I share and someone else finds it, then I lost my work and I'm not going to be credited for my work, and I'm not going to be able to apply for grants if I don't have the papers with me as a first or last author. So it's the way that our system is set up, kind of put the research in this protective position. But thankfully, in the last 10 years, it has changed a lot, and people are more and more just recognizing that if they don't share, they won't solve anything. So it doesn't matter. So it has changed a lot, thankfully, but we still have a lot to do about it. And then also, it's not only, oh, the patient wants to share, here it is, you can put it out there. Sometimes the institutions will just not let us share. Even if the patient says, I want to share, the institutions will have many rules that we have to go through to be able to share the data, and what data to share and how to share. So that was another thing, like how to create a system that gets around most of the IRB rules in a way that we still keep our patients' privacy and we keep their information protected. This is the IRB goal, right? It's not that IRB is being mean. They're trying to protect the patient. Sometimes the patient's saying, yeah, go ahead and share, but they don't even know what could be, that could happen with like sharing the data without any restrictions and in any way without protections. So the IRB is there to kind of protect the patient, and we have to work with the IRB to make sure that what we are doing is going to be beneficial for the patient and not harmful. So thinking of it, many databases out there have found a way to share different kinds of data with different group of people in different ways. I put here two that are the extreme, more or less. The first, the restricted NIH databases. These are databases that are funded by NIH that get all the raw data, mostly of the projects that are funded by NIH. And they have this data in there, and a researcher who does goes through a process of getting credentials with them have access to, so they can also investigate that data. So if I have a project and I sequence 100 patients with disease X, now I can put this data in the NIH. And if another researcher goes through all the credentialing and has a project that is fair, that is going to went through IRBs and is approved, they can also have access to analyze the same data. So it's like NIH is not spending money to get another 100 patients sequenced by other researcher when I already sequenced the 100 samples and the data is already there and can be looked at by someone else. So this is something that is very restricted, very specific in data that is very complicated to deal with. And then we have the public variant-based databases. In these ones, you are actually getting the data already processed. So you already got the sequencing, you already looked at the variants, and you kind of like spit it out what it is that this population has, and mostly giving us information about the frequency of these variants in the general population, but also sometimes classification of these variants by labs, clinical or research labs that have looked at these variants, seen these variants, classified them, and now we can know that this variant was already seen and classified by someone else. And if I find that in one of my patients, I can try to more easily connect that variant with the phenotype of my patient. So what has been missing in these databases, the first one is simplicity. Not everybody can just get to the data. And when they get to the data is not processed. What do you do with it? In the second level is phenotype. We really don't have in most of these databases that already process the data and are giving us minor allele frequency and classification, we still don't have a lot of phenotype information about the individuals that have these variants. And we don't have detailed family history. And we also don't have the capability to go back to the individual that has this variant and investigate something that was not investigated before. So when I see a variant and someone tells like, oh, my patient doesn't have a heart anomaly. And I say, well, did you do an echo? And they say, no, I'm not sure the patient doesn't have a heart anomaly, right? But if I don't know where this variant is coming from and I cannot go back to the patient, I will never know. So these are kind of things that when we tried to come up with a way to share data, we said, let's focus on that. How can we get around that? And how can we get this information when we are sharing our data without going like against the IRB rules and the privacy of the patients and trying to protect our patients as much as possible. This is when we decided that whatever we created, we would focus on sharing as little as possible, but sharing what was essential to start a productive conversation. So we decided that if we share the gene name, that would be the best way to go. So I can say, okay, I did the analysis of my whole exome sequencing on my patient, and of 80,000 variants, I found that 10 different genes are the most likely to be pathogenic. I don't know which one of them, but if I share the name of these 10 genes with the world, and someone else says, I also have a patient with variant in one of these genes, and we can talk, we can now compare the phenotype of our patients, the kind of variants in the gene, and we can go and have a further conversation about that information and try to actually define, is it the gene that's causing the disease in my patient or not among these 10 that I had initially. To make sure that the researchers would feel safe and not feel like they would be scooped, someone would just see what they had and run out with it and write the paper, we said, if the information is given to everybody at the same time, the same information, then nobody's going to be ahead of anybody else. Everybody knows about the same matching at the same time. So this is when we created GeneMatcher. It was in 2013. We decided that we would put our candidate genes for each one of the patients that we had investigated, and we wanted other people to do the same. And then the database would connect the people who had selected genes that were the same and would let them know at the same time so they can now talk to each other and decide, okay, yes, this is a good gene for this phenotype that I'm investigating. So it was created to connect not only researchers, but also the patients, the family members, the clinicians, that's many times are the ones who have the candidate gene in hands because it came from a clinical lab and doesn't know what to do with it, and the researchers. So the data that goes in GeneMatcher is mostly de-identified, right? Because you're putting the name of a gene. I cannot connect gene X with anybody. Everybody has mutation in every gene. So the fact that I found this gene as a candidate for my patient doesn't identify my patient in any way to anybody. The automated matching, like I said, if I match with Christy, both of us will receive an email at the same time telling us to each other what our email contacts are, what's the gene that we are interested on that is a match, and now we can contact each other. Some people sometimes decide that they don't like each other, they don't want to talk anymore, but they know about the match and they know that they didn't want to talk to each other. It happens very few times, but most of the times people just, even without knowing each other, they contact each other and say, like, I have a patient, my phenotype that I'm investigating is that, and the variant that I have is that, and what about yours? And then they start a conversation and a collaboration. Later, we actually decided we would match not only on genes, but also on variant. So do the patients have the exactly same variant? And this is based on genomic location information. Features, like my patients have microcephaly, short stature, and microtia. Let me find other patients around the world that have the same. Or disease name. My patient has a syndrome called Dubovitz. Let me find other patients around the world that have Dubovitz. So you can do that in GeneMatcher also. These are the numbers in the last 13 years. As of July 1st, 13,000 genes were uploaded in GeneMatcher by almost 12,000 submitters from 93 countries. And of these almost 14,000 genes that are there, about 8,500 of them have matched. So still a lot of genes have not matched, but many of them have matched. These 63% of the genes in the database have matched at least one time. Who is using GeneMatcher? And this is what Christy and I talked in the past. GeneMatcher was created for everybody, but we have not focused on advertising that to patients. I did have a patient who contacted me initially, and she helped me to actually write the FAQs in a way that would be understandable to most of the patients. But after that, we didn't really go out advertising GeneMatcher to the patients. They just learn in different ways and they come and create an account. But I think this is something that we would like to improve and actually get the word out for the patients more, because it could be helpful for them to have a tool where they are looking for researchers or clinicians or even other patients that are interested in the same gene that are mutated on that exome or the genome. We have almost 8,000 of the users in the GeneMatcher are researchers. 5,000 of them describe themselves as healthcare providers, so physicians, counselors, nurses. 635 describe themselves as patients. And then, like, I believe few of them are researchers, but not connected to human disease or human patients, but with model organisms. So 323 of these genes in GeneMatcher are actually genes that were found as candidate in a model organism that's not a human. Most of them mouse model, but also zebrafish, fruit fly, yeast, and a few others. Why is it important? Sometimes, if I find a candidate gene on a patient of mine that's causing phenotype X, and I can show that the mouse model with a mutation in that gene also has phenotype X, this is what I needed to say, okay, this is the gene causing the disease in the patient that I'm investigating. So these are the users in GeneMatcher. How has GeneMatcher helped? As of last week, GeneMatcher had been acknowledged in about 484 publications. So these are papers that are describing novel disease genes, novel diseases, novel disease gene correlations, and they're saying that that work was possible because of matches that happen through GeneMatcher. So we have here analysis that we did of what kind of genes are they. We did that in June 2020. And at the time, we had 302 genes described in 249 publications. So you can see that this number in a year went from 249 to 484 publications already. But of these 302 genes, most of them were causing autosomal dominant diseases, and then 105 autosomal recessive, 24 autosomal recessive and autosomal dominant, and a few of them causing X-linked phenotypes. Most of the genes had been GeneMatcher for more than 1 year before they were published, 221 of them, and 231 of them are now in OMIM as a disease gene. So you can see here that some of them, a few of them, already had a OMIM phenotype before they were put in GeneMatcher. And this happens because sometimes people are trying to find a second phenotype related to a gene. So the gene is already known to cause a disease, but now I think it can cause a different disease also. We do think that there are actually more publications that were enabled because of GeneMatcher matches, but it's very hard for us to track because the only way we have to track is if the paper is acknowledged or if GeneMatcher is acknowledged in the paper. If they don't acknowledge it, we can't really just track with our search, automated search. So just towards the end already, GeneMatcher started in 2013. The other database that was starting doing similar work at the time was Phenome Central from Canada. And then at this time, we were in a meeting and we talked like, doesn't make sense to have two competing databases for the patients to have, or the users to have to go to two different databases to do the same work. Why don't we just create an API that connects the databases? And then if patient or a user goes to GeneMatcher, they can also search in Phenome Central and vice versa. So we created a group called Matchmaker Exchange, and we created an API to connect databases that were doing the same thing we do, maybe in different countries, maybe in the same country, but in a slightly different way. So we have now here, you can see that we have 8 different databases that are all connected. So if you go to GeneMatcher and you make your search in GeneMatcher, you also can actually send your search to any of these other ones here. And Phenome Central is in Canada, MyGene2 is US, RD Connect is Europe, IRUD is Japan, Seeker is in US, Decipher is Europe, and Patient Matcher is Europe. So you see that you are from GeneMatcher not connecting to 11,000 users around 93 countries. You can also go to different databases that have data from research that is being done in different countries around the world. So that has worked well. But we still have many improvements that we are working on now. And I talked to Christy, I talked about writing the grant and trying to get more funding to keep the work going and improve. And sounds like we are going to get it, cross fingers that it's really happening. But we are going to keep improving it. And I think talking to the patients and getting the patients to participate more can help us to improve in a way that it makes it also more useful for them. Like right now, we have been talking a lot with the model organism community because we want to make it more useful for them also. So we want to be talking with different group of users that can give us an idea of how it can be more helpful than already is and how we can improve the work that we are doing. So I'm very happy that Christy brought me here today. I was very happy to hear your background because you are exactly the group of people that we would like to serve better. So I welcome any comments, suggestions, questions, and you can always contact me through email also later. Thank you very much.
Wonderful. Thank you so much, Nara. We had some comments in the chat, and then I will let anyone who would like to answer a question maybe unmute themselves. But Winton had to run, but she said this is fascinating. And Lourdes had a comment, this is very interesting information. Thank you. So just wanted to pass along those comments. Did anyone have any questions that they'd like to ask?
I have a question. This was fascinating, thank you. And I love that it's API based and that you're inviting other organizations to be a part of this. You know, it's funny, as you were talking, RareX popped up and then RareX is another database that is working with rare diseases. And how do you create, with the API, I think you're creating a sense of collaboration, but how do you work in encouraging other companies with disparate databases? I mean, we're only as good, everyone kind of has to invest in this and be able to exchange information openly. And how do you combat that, for lack of a better word?
So to be very honest, like NIH had a big part on that. They started making data sharing a big requirement of any funding that they are giving away for sequencing. So, you know, as I said, researchers are a little bit reluctant to let the data go. And also the institutions are very worried about being sued if something happens that a patient of them didn't like and the data went out through their institution. But NIH has come out as a big supporter of data sharing, and most of the sequencing that is done in nowadays with NIH funding has to be shared one way or the other. So at least in US, it has been NIH, a big, I think has done a big push. Around the world, in Europe, it's just the need. You know, like if you don't share, you really don't solve.
Right.
And people got more and more aware of that with time. It's like, doesn't matter if I have all this data here, sitting here, like I'm not going to solve if I'm not going out and sharing and putting people to talk to each other and collaborating, and not only in the same country, but different countries. So you see that there are some people from Europe, 3 different groups in different countries. And then we have the Japan group, and Canada was one of the beginning with us. So I think it was part a need to get the case solved, and then part the funding institutions should say, like, you have to. It's not an option.
I love that the NIH stepped up like that.
Yeah.
Thank you, Heather. Great question. And Hanan has her hand up. Hanan, would you like to unmute and ask a question?
Yes, hi. Thank you so much for that information. There were some things that obviously are new to all of us. So as you were speaking, I had Google on the side, Googling things up to understand what some things that you were mentioning. But 2 really ideas popped into my head. Number one, and I'm probably sure that some of this question could be probably answered with family genetic testing. So when my daughter, for example, was diagnosed, we had no known family history. And so, and you know, my background or where I'm from, I'm originally from Ethiopia. So in the African world, in general speaking, there's a huge problem in terms of not having, you know, like, for example, when the question was asked, do you have a family history of any kind of genetic disorders? Or do you have any family history of, you know, certain diseases and things like that? So that's, you know, data is really non-existent. So it's really difficult to find out if there was anything in the family. I know of, like, my husband's cousin, for example, had a disorder and nobody knows what it is. And I've been asking around to see if there was any kind of lineage there connecting to my daughter to hers. Even then, like, this is close family that I can't even find answers for. So when you were talking about this database and things like that, it was just kind of, to me, it felt like my background is so behind on this. And what can we do, or how can it be that, you know, those of us who are of African descendants can also get on this and find ways that maybe we can have answers for families, our daughters, our kids, and things like that. And then the other point that I wanted to mention, I really couldn't help but think of the story or a book that I recently read for my tiny little neighborhood book club called The Immortal Life of Henrietta Lacks. And when you were talking about your gene, what's the word? I'm sorry, you just mentioned it. I can't think of it. It got me thinking of the story of the whole HeLa cells and how, you know, so many things have moved from, you know, not sharing information and things like that. But this is more of a comment, not a question. But I just wanted to share with you guys that story really related, for me at least, when it came to you mentioning about having access to the genetic information or the genome information. So then my question, or at least maybe comment, I'm not sure if you would be able to have that answer right now, for developing world who do not have access to this amazing, you know, technology or even the research and the information. It's difficult for people like me to kind of even have any idea of family history of any kind of disorders or diseases. So thank you for your time, and thank you for the question, the opportunity to ask the question.
Of course. So the one thing that we tried to make with GeneMatcher was easy enough that everybody could use it anywhere. So the 93 countries that use it include many countries in Africa also. And you don't need to have much more than the gene name, because once you're connected, then you start the conversation. So if I'm talking to a doctor who actually put the gene there for you, or even with yourself, now I can actually talk to you about it and come up with the questions that are important and relevant to try to connect this gene with the phenotype. So you don't need to put that information in the database right away. This is one way to get around, I don't have the information. So if I don't have the information, I can't participate. No, you can. You participate with the gene name, and then we can start talking about what all the information we need and how to get it. And if we don't, we don't. But if we do, we get it and put it together. So that is the way that we try to get around that and make it as accessible as possible for people in low and middle income countries, and also to people who don't have a lot of information themselves about family history or even about the phenotype itself. I study a group of diseases that the patients have cartilage tumors, and sometimes they only know that they have the cartilage tumor in the hand because the doctor never asked the x-ray of the whole body. And then they don't know that they have the cartilage tumors also in the leg, in the spine, everywhere else. So sometimes we don't know information about ourselves. We try to get around that by not asking so much upfront.
And just a clarifying question for that, Nara, can you start, if you don't have the gene name, if you don't know what the gene is, can you start with phenotypic data?
Yes. So you can go to GeneMatcher. Yes. You can go to GeneMatcher and say, I want to find all the researchers or all the clinicians or other patients who have interest on disease X. So you can do that also. And then if someone else in there is studying Dubovitz, for example, that we don't know what the gene is that causes Dubovitz, but your interest is on Dubovitz, then you find other people with interest in the Dubovitz also. And then you can talk what interest is and how you want to solve it and what you want to do about it and go from there.
Thank you. And we have about 2 minutes left, so I'm going to take everything else.
I hijacked your meeting. I'm so sorry, Christy.
It's okay. We have one more hand up, which is Trish Thomas. Trish, I'm so glad to see you on here. Would you like to go ahead and ask your question?
I would. Thank you very much. My question is, are you connected with the Indian Health Service, who provides health care to American Indians in the United States?
No, I'm not. I would be happy to connect with them. If you know someone who we should be talking to, I would be happy to, but we are not.
Okay. I can probably get Christy the contact information for the IHS, the Indian Health Service Director at the federal level. And because I think it is important, and a lot of, we have pockets of children and adults who have very specific genetic conditions like Down syndrome. There's a whole village that has quite a few children. Again, and the families don't appear to be related. We have Cornelia DeLange and other types. And I've been working with the Navajo Nation, but because of the sovereignty, especially with our 19 Pueblo tribes in the state of New Mexico, we're unique in the sense that we've never become U.S. citizens. We've always just been here. We didn't migrate from anywhere. But other nations, migrated from different places, either through Canada or up through the South. I know that there's quite a few different types of genetic conditions, but because of, again, the culture and tradition, it's so hard we try and get information to the families, but because it's considered taboo, they don't speak of it as a genetic condition. So the terminology has to be kind of not used, say in the, like if you're a geneticist, you will have to change your type of terminology and not use the word genetic, but to describe the condition itself. Mm-hmm. That is acceptable. But I will give Christy that information and hopefully she can, she'll get it to you and you can see how that might work and explain the program to them. Because if families are more concerned, again, depending on the tribe they come from, about how the information is going to be used, will their names be made public and stuff like that. But I will stop now because I know we're almost done with our time. Thank you.
Thank you, Trish. I would love if you could pass that along to me and I'll get it to Nara. Thank you.
Okay.
Great. All right. Well, just to wrap up, thank you for those of you who have stayed on for a few minutes. Just to wrap up, I just wanted to give you just 3 reminders. I'm going to skip over the red flags and the explainer video, but I'm going to send those out in an email with the links. I've already sent the red flags document, but I would love some feedback on this explainer video intro. So I'll send that out on email to you all. And then just 3 reminders going forward into our next quarter. We have our Genetics Summit coming up on November 9th and 10th. Registration is going to be opening in August, and it is there is no charge for this event, this virtual event this year. So we would love our genetic ambassadors to help us spread this word far and wide once registration opens up. The next is you've heard a couple genetic navigators introduce themselves. They are up and running. They have email addresses that are working, and they are starting to navigate with families this month. And so when I post this slide deck, it's hyperlinked. You can go to their biographies there at that link and their email addresses. And then last but not least, we have our Time for Genetics cohorts. If you remember, this was our educational efforts for pediatricians and primary care practices in genetics. And we are opening up cohort number 2. And so the link for that application is live and will be linked in this PowerPoint. And you can share that if you know a clinic in your own state, maybe it's your own pediatrician, your own primary care doctor that you would like to tell about that program, you can share that link with them. So that was a quick run through. We're a little bit over time. So with that, I'm going to wrap up and close the meeting, and we'll catch up in a couple more months. But I just want to thank you, thank our guest speaker, Nara, today for the great information she shared with us. And if anybody's interested in contacting her and talking with her more, I reached out through the GeneMatcher website, but I'm sure she would be happy to correspond by email as well. If you need an introduction, reach out to me.
Yes.
Thank you, everyone.
Bye-bye. Thank you, Christy. Bye. Sorry again. Thank you.
Oh, it's okay. Thank you, Nora. We appreciate all the information you shared. Thank you so much. Thanks, everyone. Bye-bye.
Thank you. Bye-bye.
Bye-bye.